All right, welcome back to another video. So this week's topic is on gas transport. So this is a fairly lab-heavy week, not very lecture-heavy. So this lecture video is going to be fairly short compared to previous weeks. So one of the things that we're doing today is we're going to be measuring the amount of oxygen that's being transported or that's available to be transported throughout a body. So I'll go through a little bit about the experiment, but before you come into the lab, make sure you have read the lab manual, you've watched this video, taken the lab quiz, and if you have a pair of goggles, bring those goggles. If not, we have a box of crappy ones that you can borrow. We'll be doing a couple different calculations in lab, so be sure to bring either your phone or a calculator, something that has, or that you can multiply, add, subtract, divide, stuff like that. So what we're doing today, or Wednesday or Thursday, is we're measuring the amount of oxygen that's available for a respiratory pigment called hemoglobin. So before I get into hemoglobin or other respiratory pigments, I'll go through why we have respiratory pigments. I'd love to harp on the surface area to volume ratio that's really driving all factors, such as size of organisms, the stuff like that. The examples are escaping my mind right now, but that same surface area to volume ratio that I keep talking about, which you guys are probably fairly annoyed by now of me mentioning it so often, is that it affects the respiratory functions of these organisms. So when you're extremely small, you don't have to have fairly complex respiratory systems, because oxygen and CO2 will diffuse into your body or out of your body through your cuticle just passively. So you don't actively have to pump oxygen into your body and then transport it around to all the cells that need it. You just have to basically sit there and then allow oxygen to diffuse in and CO2 to diffuse out. So in these organisms that are fairly simple, so if you think of like the platyhelminthes, so those planaria that we're looking at, they don't have a circulatory system. They're not pumping blood or hemolymph or these fluids throughout the body, because they're just relying upon the passive diffusion of oxygen or CO2 out of their bodies. And so these organisms have fairly low metabolic rates. They're not, sprinting hundreds of meters to catch a prey organism. They're just slowly kind of meandering through a substrate or through water in search of decomposing organic material, other slow-moving organisms, or plants. So they don't have these super high metabolisms, and they are small enough where they don't need a complex circulatory system. But as organisms are getting larger, they're getting more complex. They can no longer rely on the diffusion of oxygen passively from the external environment to the cells that need it most. So as these organisms are getting larger, the amount of oxygen that's diffusing into their bodies just through their skin doesn't penetrate enough for it to get to cells that need it most. So if we didn't have lungs and we relied solely on the diffusion of oxygen into our bodies, we'd be dead, because the amount of surface area that we have is fairly low compared to the volume inside of our bodies. So instead we have very complex circulatory systems and respiratory organs that we use to pump oxygen into our bodies, and then CO2 out of our bodies. But since we can't just pump oxygen or air throughout our bodies, because once again we would just be pumping, we couldn't pump enough air into our bodies and out of our bodies fast enough to transfer the amount of oxygen that we need. Instead, we have these respiratory pigments. So there's hemoglobin, hemocyanin, there's another hemo, and then there's cyrocutin. So there's a bunch of respiratory pigments. The major ones are hemoglobin, hemocyanin. And so these pigments have an extremely high affinity for oxygen and CO2. So they are actively grabbing oxygen from the air at rates higher than other fluids such as water or I don't know, other interstitial fluids. So we have these respiratory pigments solely for the purpose of grasping oxygen from the air and transferring it to the cells that need it. So we have, chordates have the respiratory pigment hemoglobin. Invertebrates, some invertebrates have the respiratory pigment hemocyanin. And I'll talk about that a little bit more on the next slide. So there's a couple different methods for organisms to transport those respiratory pigments throughout the body. In extremely small organisms, ones that usually are less than a millimeter from that cell wall or from those cells to the actual outer cuticle, they rely on cutaneous respiration. So they're not actively pumping blood or hemolymph or this interstitial fluid throughout their bodies, and they're not really carrying these respiratory pigments because they can rely solely on just the passive diffusion of oxygen into their body and then carbon dioxide out of their body. When we start having organisms that are a bit larger in scale, such as invertebrates like the flatworms, so platyhelminthes, or insects, all the way up to gastropods and mollusks, you start to have open circulatory systems where you have the active pumping of some sort of fluid carrying respiratory pigments throughout the body. So in invertebrates, especially those with an open circulatory system, they're using hemolymph as that fluid, and so this has one of those respiratory pigments dissolved inside, and it's using some sort of surface such as gills or hook lungs or tracheals to carry that oxygen directly from the outside air and then carry it to cells that need it most. So those that are deeper in the body, muscles, to supply the nervous system, stuff like that. We have larger scaled organisms, especially those invertebrates, so we have some sort of backbone, such as mammals or other chordates. Those respiratory pigments are packed into what we call erythrocytes. So those erythrocytes are red blood cells. In our own blood or body, blood is composed of three different, I guess you consider it cells. So you have the leukocytes, those are the white blood cells. You have erythrocytes, which are these red blood cells, and then you have the platelets. So we're going to slowly focus on those erythrocytes, and so those are the red blood cells. So in humans, our blood's red, and it's red because we have hemoglobin. So this is a iron-containing protein that has what are called heme groups. So those heme groups are where oxygen and CO2 are binding to. So in our lungs, we're pumping oxygen into our body. So directly across from our alveoli are capillary beds. So oxygen is diffusing into these capillary beds and directly into our erythrocytes, where these heme groups are basically grasping onto these oxygen molecules. So as that blood is then pumped down the length of the body to whatever cells need it. So if you think about when you're running, you're using a lot of oxygen, so that blood is going to be pumped down to where it's needed most. So if you think about your legs are going to be using oxygen and then releasing CO2 just through cellular respiration. So as that blood that's carrying oxygen approaches those cells that are, in high demand of oxygen, they release the heme groups release that oxygen due to the differences in pH that they encounter, and then they become much more what's the word for it? They have a much higher affinity for CO2. So they then grasp, those heme groups then grasp onto these CO2 molecules, and then the circulatory system transports it around to the lungs, where we then exhale and release that CO2. So in most invertebrates, well, not most invertebrates, in crustaceans, mollusks, and horseshoe crabs, so class Meristomata, they have hemocyanin instead of hemoglobin. So hemocyanin has copper in its heme groups instead of iron. So that copper acts in the same exact way. So this is that copper acts as a binding site for oxygen and CO2. So that copper grasps onto a oxygen molecule near the gills or the book lungs or whatever structure that organism, that invertebrate has for basically respiring. So that copper grasps onto the oxygen, it's then transported to whatever cells need it most, it releases oxygen, and then the difference in pH causes it to grasp onto a CO2 molecule. From there, it's then transported back to the gills, where it releases that CO2, it picks up the oxygen molecule, and the cycle repeats itself. So one way you can just visually tell the difference between hemocyanin and hemoglobin, or organisms with hemocyanin or hemoglobin, is just by looking at the color of that that blood or that hemolymph. So organisms with hemocyanin will have blue blood, just because the presence of copper inside of that hemolymph. So you probably have seen this picture before. I used it on the last lecture that I gave. So this is a horseshoe crab being drained for LAL, so Limulus amoebocytis lysate, something like that, which is found in the blood of these horseshoe crabs. So that's all that blue that you see are is that respiratory pigment, hemocyanin, which has copper. So on the bottom left, you'll see a vertebrate and an invertebrate. So there's a glass frog found, I think, in Brazil. So this organism has hemoglobin inside of its erythrocytes. So those red blood cells are carrying hemoglobin, so that iron-rich protein that's colored red because of the iron. So a little bit of stream ecology for you all. This is this organism that looks kind of like a segmented worm is actually a larvae for a non-biting midge. So it's in the family Chironomidae, and so they are pigmented this way because they just have a high concentration of hemoglobin throughout their body. So these organisms typically live in the hyperrheic zone or the benthic area of a stream. So that means basically underneath the stream bed where there's still water, but there's not water flowing. So these organisms live in fairly anoxic or anaerobic conditions. So they have to be able to absorb as much oxygen as they possibly can from those fairly low oxygen environments. So they have tons of hemoglobin throughout their body, which gives it this red, which is why it looks so darkly red. If you ever find, or if you start doing stream surveys, or if you are working with Department of Natural Resources, and you're going out and surveying streams or lakes, and all you're finding are these chironomids, or these larvae, that's probably a bad sign for the site that you're sampling, because these guys can live in areas where there's low oxygen, which is the characteristic of streams or lakes that have very poor water quality. So these organisms, they're taking advantage of that niche, that low oxygen, fairly anoxic or anaerobic environments, so they can thrive in these areas with, well, low oxygen. So yeah, so this is a hemoglobin molecule. So this is what we'll be doing in lab. We'll be dealing with hemoglobin. Specifically, we won't touch hemocyanin just because hemoglobin is much easier to find. So this is the general appearance of a hemoglobin protein. So as you can see, it has 4 heme groups. Each of these green circles they see are where oxygen or CO2 is attaching. This really isn't a great diagram, basically because these heme groups are actually colored red because of the presence of iron, and then the actual structure of these beta molecules or alpha molecules, these polypeptide chains, they're actually colorless. They're just dyed this way or colored this way so you can see compared to the heme groups. So in reality, these heme groups are what actually gives hemoglobin its color. So in blood, that color is from these heme groups, not these polypeptide chains that you see in this diagram. So this is the protein that's responsible for the transportation of oxygen throughout our body, and then transportation of CO2 to our lungs, where we can then exhale and get it out of our body. So one of the ways that this, one of the neat, aspects about this is that these polypeptide chains and heme groups are fairly susceptible to changes in pH. So as CO2 is being released into our blood, that changes the pH of our blood, so it changes the acidity, which causes these heme groups to release oxygen and then collect CO2 or carbon dioxide. So that's one way, or that is the way that these hemoglobin molecules transport oxygen to our cells and then collect CO2 and then transport it away. So one of the weird things about hemoglobin is that while it does have a fairly high affinity for oxygen and carbon dioxide, it has an even higher affinity for carbon monoxide. So this is a byproduct of combustion, and so this is why it's important for you all to have like a carbon monoxide detector in your homes or your apartments, because it does attach to these heme groups much more readily than carbon dioxide or oxygen. So as you're breathing or breathing in carbon monoxide, it's replacing oxygen and then it's not being released. So more and more of these hemoglobin proteins or the blood cells are absorbing carbon monoxide, but they're not releasing it. So in essence, you are slowly reducing the amount of oxygen that your body can handle or intake, and so you end up, or this ends up being fatal, which is carbon monoxide poisoning. All right, so the way we measure the amount of oxygen that's available in the air for our bodies to diffuse, well, into our cells is through what's called partial pressure. And so this is a measurement or more of a proportion of oxygen within the atmosphere at a given pressure. So that proportion is generally 21%. So out of all of the gases in the air, only 20% of it is actually oxygen. Well, actually less than 21%. It's roughly 20.95%. The rest of it is mostly nitrogen. In fact, I believe there's only 1% left over, and so that 1% is containing CO2, carbon monoxide, methane, hydrogen, helium, stuff like that. Oh, and water vapor. So not technically a gas, but it's still present in the atmosphere. So that percentage or that proportion is then multiplied by atmospheric pressure, at whatever level or elevation that we're at. So since we're roughly around sea level, we're just going to use sea level as a baseline for our measurements. So the atmospheric pressure at sea level is roughly about 760 millimeters of mercury. So that unit, millimeters of mercury, is measuring the amount of pressure that it takes to push mercury up 1 millimeter in a J-shaped tube. So at sea level, the amount of pressure that atmosphere is exerting on a single point is pushing mercury up 760 millimeters up one side of a tube if it's exerting pressure on the second side of that tube. So kind of complex way to picture it, but it's one of the easier ways to measure atmosphere or the pressure in the atmosphere without having to do lots of conversions. So to just do some simple math, we're just going to take that percentage of oxygen that's available in the atmosphere, and then we're going to multiply that by the pressure that's exerted at sea level. So even though we're not at sea level, we're roughly about, 150 feet above sea level, we're just going to assume that we're at sea level for all intents and purposes. So we're going to multiply that percentage, so 20.95%, and we're going to multiply that by 760 millimeters of mercury. So that's going to give us roughly 159.6 millimeters of mercury, and so that's the partial pressure of oxygen at sea level, and so that is the amount of oxygen that's available to our bodies while we're breathing. So that's the amount of oxygen that's available for us to actually transfer to our cells. So that number, 159.6, is fairly less the amount of oxygen that's available outside of our bodies. So once we breathe in and we have that oxygen inside of our lungs, that number drops drastically. So we're only replacing about 20% of the air in our lungs at a single time. So the amount of oxygen that's available in our lungs is drastically lower than in the outside air. So that partial pressure of oxygen from the outside air to the inside of our lungs is fairly different. So in lab, what we're going to be doing is we're going to be measuring we're going to be changing the partial pressure of oxygen by adjusting the pressure within a volumetric flask. So we have a vacuum pump in lab, and we're going to be using it to slowly drop the level or the pressure within a volumetric flask, and then we're going to be measuring the amount of oxygen that's saturated inside of these hemoglobin, or these heme groups inside this hemoglobin. So we'll go through several steps, several calculations while we're in lab. If you have your own goggles, be sure to bring them, because we are dealing with glass that is susceptible to shattering. And if you have a calculator, bring it. Your phone will work just fine. So we're making a oxygen saturation curve, and so we're going to be plotting the saturation of oxygen at different partial pressures. And so the way we'll be doing this is we'll be taking the transmittance of that hemoglobin, this liquid with hemoglobin inside of it, and we're going to be comparing that to saturation of oxygen. So one of the ways we'll be doing that is we'll be taking measurements of light. We'll be shooting light through this volume of hemoglobin. We'll be measuring transmittance, and then applying that to a saturation curve, and then applying that to partial pressure. So there's several steps we'll be making in this lab. In fact, one of them involves chemicals. So we're dealing with sodium hydrosulfite. So one of the things that we can't do with a vacuum pump, it's not strong enough, is we can't actually create a vacuum. We're just reducing pressure. And so what we'll assume when we get down to extremely low partial pressures, or pressures within our volumetric flask, we'll assume that we'll have 0% partial pressure within within our volumetric flask by adding the sodium hydrosulfite. So that sodium hydrosulfite is going to absorb as much oxygen as it can. It has a much higher affinity for oxygen than hemoglobin does. So it'll basically remove all the oxygen within that volumetric flask. And so at a partial pressure of 0, or atmospheric pressure of 0 in a vacuum, we could assume that there isn't any oxygen available for the hemoglobin. So we'll have a baseline at 760 millimeters of mercury, so sea level. And then we'll have a second baseline at 0 millimeters of mercury, so at no pressure. But since we can't really get to 0 millimeters of mercury, we'll be using that sodium hydrosulfite to kind of adjust our data a little bit. So yeah, that was kind of a side note, something I usually bring up in lab. But just make sure you're watching videos, take the quiz. Like I said, if you have a pair of goggles and you want to bring them in, go ahead and bring them in. If you bring in a calculator or your phone, yeah, that should be about it. So I know this week's video was pretty short compared to most others, but it's almost like a break after the midterm. So I'll see you guys in lab on Wednesday or Thursday. See you then.